Hello students, today we are studying with biology chapter number 7 cell division. We have already started with this chapter in our last lectures. Till now what all we have studied? We have studied about the process of mitosis, we have studied cell cycle, okay, and we have also started with the process of meiosis. Okay, the introduction part of the meiosis was done in our last lecture. So let us continue with this chapter. What is meiosis? As I have already discussed in detail, but let me revise with the definition of this meiosis. The form of cell division by which gametes with half the number of chromosomes are produced is known as meiosis. Okay, the process by which the cell division by which the gametes with half the number of chromosomes are produced is known as meiosis. Okay, we have studied about meiosis in detail in our last lecture, introduction part. There are some phases of meiosis. Let us see the phases of meiosis. Meiosis 1, it is also known as reductional division. Okay, and it produces diploid mother cell to produce two haploid cells. It consists of following phases, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 is divided into 5 substages, that is leptotene, zygotene, pachytene, diplotene and diakinesis. So when I say prophase 1, there are 5 substages. Okay, so today we are going to study about all these 5 substages in detail. Then in our next lecture we will be studying metaphase 1, anaphase 1 and telophase 1, and after that cytokinesis 1. So let us start with prophase 1 today. Prophase 1 is the longest and the most complex phase. Okay, prophase 1 is the longest and most complex phase. You can say 90%. Okay, chromosomes are condensed in this phase. Synapsis occurs, that is homologous chromosomes come together to form tetrad. Tetrad is 2 chromosome or 4 chromatid sister and non-sister chromatid structure. What is tetrad? It consists of 4 chromatids, that is sister and non-sister chromatids, and it is having 2 chromosomes. So this prophase 1 has longer duration, and the significant features which are peculiar to meiosis occurs in this phase. This phase can be divided into 5 substages, that is leptotene, zygotene, pachytene, diplotene and diakinesis. So one by one let us study these substages, that is first one, leptotene. What do you mean by leptotene? It is the first stage of prophase 1 and is called leptotene or leptonema. Okay, first stage of prophase 1 is also called as leptotene or leptonema. All the chromosomes begin to condense, so they become visible as fine thread. Okay, in this phase what happens? All the chromosomes are beginning to condense, and because of that they become visible as a fine thread. There is marked increase in the nuclear volume. Okay, there are marked increase in the nuclear volume. The process of homology search, which is essential to the initial pairing of homologous, begins during this phase. So the synaptonymal complex is formed during this zygotene stage. At the completion of zygotene, the paired homologs take the form of bivalents, and the number of bivalents in each species is equal to the haploid number. So I repeat this protein stage. In this what is happening? The volume of nucleus increases, the chromosome becomes distinct, long, thread-like and coiled. They take up a specific orientation. Okay, called the bouquet stage. The specific orientation is called as the bouquet stage inside the nucleus. This is characterized with the ends of chromosomes converge towards that site. Okay, so of the nucleus where the centrosome lies. Okay, The centriole divides into two and migrate to opposite poles. So this is all about the leptotene stage. Let us move forward with zygotene stage. This is the second stage, the synaptonymal. Okay, the synaptonymal complex is formed during the zygotene stage. At the completion of zygotene, the paired homologue takes the form of bivalence. Okay, the paired homologue is taking the stage of bivalence. And the number of bivalents in each species is equal to the haploid number. So, the zygotene stage, also known as zygonema, this stage begins with the initiation of pairing between homologous chromosomes and it ends with the complete pairing. The process of pairing at the end to end between the homologous chromosomes is known as synapsis, that is homologous dyads. Okay, I repeat this: zygotene stage. The synaptonymal complex is formed during this zygotene stage, and at the completion of zygotene, the paired homologue takes the form of bivalents. The number of bivalents in each species is equal to the haploid numbers. Okay, so what is zygotene? It is an intimate pairing of non-sister chromatids of homologous chromosomes. Takes place by formation of synaptonymal complex. This pairing is called synapsis. Each pair consists of a maternal chromosome and a paternal chromosome. Chromosomal pairs are called bivalents. Okay, chromosomal pairs are called bivalents or tetrads. Next stage is Pachytene stage. What do you mean by Pachytene? The Pachytene stage, also known as Pachynemia. Okay, Pachytene stage is also known as Pachynemia. The process of synapsis is complete in this process. That is, in this stage, that is Pachynemia stage. The two haploid homologous of each bivalent appears to be attached with each other, and one or more points their attachments are known as chiasmata. Wherever there will be the attachment of the chromosome, the point over there will be known as chiasmata. Crossing over is a precious break and swapping and reunion between two non-sister chromatids. Okay, crossovers make a easy and a new gene combination, and which are an important source of genetic variations in population. I repeat this zygotene stage. Listen carefully. Each individual chromosome begins to split longitudinally in two similar chromatids. At this stage, tetrads become free nuclear in appearance because of presence of four visible chromatids. The homologous chromosomes of each pair begin to separate from each other. However, they do not completely separate but remain attached together at one or more points, and that point of attachment is known as chiasmata. Okay, the point of attachment of chromosomes is known as chiasmata. These points appear like cross, okay, X cross, known as chiasmata, and chromatids break at these points and broken segments are exchanged with non-sister chromatids of homologous chromosomes, and this is called as crossing over or recombination. This is also known as crossing over or recombination. Next point is, next stage is diplotin stage. What do you mean by diplotin stage? The diplotin stage, also known as diplonemia, DNA recombination is complete. The chromatids continue to shorten and thicken, and the four sister chromatids in a group is called tetrad. Okay, the four sister chromatids in a group is also known as tetrad. The synaptonymal complex begins to break down. The paired chromatids begins to pull apart, causing the strands to separate longitudinally, and the chiasmata tend to become terminalized as the meiotic process continues. If meiotic process continues, then there are chances of reduction. Okay, there are chances of separation. So what is diplotin stage? Though chiasmata are formed in packetine, they become clearly visible in diplotin. Okay, due to the frequency of gene. Okay, frequency. What is happening? Though chiasmata are formed in packetine, they become clearly visible in diplotin due to the beginning of repulsion between synapsed homologous chromosomes. This is called as desynapsis. It involves disappearance of synaptonomal complex. Okay. So next topic is diakinesis. That is, next stage is diakinesis. The chromosomes become shorter and thicker due to condensation. Nucleolus and nuclear membrane, nuclear envelope disappear towards the end of the diakinesis, and the spindle apparatus becomes organized during this stage. The events, the centrioles migrates away from the centriole. Okay, the spindle apparatus becomes organized and the centrioles migrate away from one another. So this is all about diakinesis. What happens in this? In this phase, the chiasmata begins to move along the length of the chromosomes from the centromere towards the end of the chromosome. Okay, the displacement of chiasmata is termed as terminalization, and the terminated chromosomes exist till the metaphase stage. Okay, the nucleolus disappears and nuclear membrane also begins to disappear. Spindle fibers start to appear in the end of the diakinesis stage. Okay, in the cytoplasm, that is in the end of the diakinesis stage. So that's all for today. Okay, thank you for today. We will continue the same chapter, same topic in our next lecture. Okay, we will stop here. Thank you everyone.